Hi everyone. First of all, thank you all for being here now, and thank the organizers for giving us the opportunity to show our study. So, before talking about the elemental diets, I'd like to give you a brief explanation about the digestive process. So the food containing carbohydrates, lipids, and proteins is firstly digested in our body by the enzymes for forming smaller parts, and those smaller parts, like fatty acids, short peptides, trisaccharides or disaccharides, they are later diffused into the cells of the intestinal mucosa. But elemental diets are already formed by those smaller parts such as monosaccharides, free amino acids, medium chain triglycerides, vitamins, and minerals. They are highly efficacious and have minor side effects. And because they are allergen-free, they are proximally absorbed, they have a low fat content, no food additives, and they modulate the microbiome, among other characteristics. They can be used to treat several GI diseases and disorders such as eosinophilic esophagitis or gastroenteritis, IBD, SIBO, and other diseases. Dr. Mark Pimentel from our group had previously shown in a retrospective study that the use of elemental diet improved the symptoms in subjects with small intestinal bacterial overgrowth, and there were no severe or serious adverse events in that study. But so far, no one had shown the effects of the elemental diet in the context of IMO in a prospective study. But what is IMO? IMO stands for intestinal methanogen overgrowth. It's associated with methane-producing archaea, such as METHANOBREVIBACTER SMITHii. The diagnosis consists in exhaled methane levels higher or equal to 10 ppm, and it's associated with constipation, bloating, and abdominal discomfort. The treatment consists in the use of antibiotics, but there is recurrence and some refractory cases, and also in the use of elemental diet. But as I told you, there is no prior prospective study. And the treatment response can be monitored by a single fasting methane measurement. On top of no prospective study with IMO, palatability is the main limitation in the use of elemental diet. So as you can see here, there is a high dropout rate in some clinical trials. So for this reason, we developed this study to assess the effect of a novel palatable elemental diet on M.smithii levels in the stool of subjects with IMO and the correlation with single fasting daily breath methane measurements. 24 subjects with IMO participated in the study, and the study was a total of 5 weeks. The first thing they did on visit 1 during the screening phase was a lactulose breath test to diagnose IMO, and they did a second lactulose breath test at visit 4 or at the end of elemental diet to assess the response to the elemental diet, but we won't focus on the LBT today. On visit 1, they also did a palatability test where they received 2 different elemental diets, including ours, and they needed to give scores for those 2 diets. They were blinded. They gave scores for taste, smell, and other characteristics, and they also answered a symptom questionnaire, and they were asked to answer the symptom questionnaire during the entire week of the screening phase. Moreover, they came back on visit 2, and they were asked to record their daily symptom questionnaires during the entire 2 weeks of elemental diet, plus 2 weeks of follow-up phase. They also received packages of the palatable elemental diet here on visit 2. They were fasting, and that those packages, the combination was, the total number of packages was according to their eucaloric needs. Moreover, they received extender tubes to collect their fasting daily methane, and on day 28 or visit 6, the treatment response consisted on exhaled methane levels lower or equal 5 ppm. And the stool M.smithii was assessed on visit 2, 4, and 6. We isolated the DNA from stool, and later this DNA was used to assess the levels of M.smithii by digital PCR. So the first thing I want to show you here is that the subjects were on average 48 years old. They were mostly female, and 22 out of 24 participants, they had a previous unsuccessful treatment. I mentioned that they did a palatability test. They were blinded for the palatable elemental diet and a commercially available elemental diet. They needed to give scores for taste, smell, consistency, and other characteristics. And as you can see here, the scores were mostly good for the palatable elemental diet, for taste, smell, and the other characteristics, and they were mostly bad for the commercially available elemental diet. So we noticed that there was a significant improvement in palatability, and all subjects tolerated and completed the 2 weeks of exclusive palatable elemental diets. Then they came back on visit 2, they received all the packages of the palatable elemental diet. Then, as I mentioned before, the response for the trial, the entire trial, was assessed on day 28 according to their fasting, their daily methane measurements. If it was lower or equal 5, they were responders. So we found 18 responders versus 6 non-responders. And as you can see here, the daily methane averages dropped below 5 ppm on day 2 and continued low throughout the duration of the study in the responders. Moreover, when we compared the levels of daily methane at the end of elemental diet versus baseline, there was a decrease, a significant decrease, and this decrease was sustained until the end of the trial. Then we assessed the levels of M.smithii in their stool on visit 2 before elemental diet, visit 4 at the end of elemental diet, and visit 6 at the end of the entire trial. We could test 23 samples in total. 61% of them were positive for M.smithii. Then we took only those positive samples, and we tested M.smithii levels at the end of elemental diet versus baseline. We saw a decrease, but this decrease achieved significance at the end of the trial compared to baseline. Then we correlated the levels of M.smithii with the daily methane measurements, and we saw a positive correlation at baseline, at the end of elemental diet, and in the reintroduction phase or the follow-up phase. Moreover, the palatable elemental diet improved overall symptoms in IMO, especially when we compare the end of a follow-up phase or the end of reintroduction of food phase versus baseline, as you can see here. However, the study has some limitations, such as the lack of a blinded placebo group, the sample size, and the short follow-up. So we conclude that palatable elemental diet was actually palatable. They all finished the trial, they completed the trial. A 2-week course of palatable elemental diet induced a sustained reduction in stool M.smithii. The decrease in stool M.smithii was positively correlated with the decrease in the daily methane measurements, and palatable elemental diet improved overall symptoms in IMO. As future directions, we have full metagenomic sequencing, microbial gene functional analysis, and microbial interaction analysis already planned. And I'd like to thank the MAS program and all the participants in the study.